Let's go ahead and get started. Thank you everyone for spending 45 minutes of your day, the trade show today up with me to talk about biocompatibility. It's kind of interesting with these lights and with this microphone. I feel like I should be on Broadway, but I promise you I'm not going to sing because that would make you guys, it would not be a good activity for us. Before we start, I just want to let everyone know I have some information up here for you guys. So I have a couple of flyers. We also have our Sotera Academy, which if you haven't gone on to our Sotera Academy, is where we put all our training webinars and videos and things up for free. And so you can kind of keep track of the latest things that are happening within just not biocompatibility, with sterilization, packaging, everything that Nelson or Sterigenics kind of talks about. We upload them up on the Academy. So there's a flyer here in case you don't know about the Academy. Probably one of my favorite things is this slide rulers. So this is the chart out of 10993-1, but it's in slide rule form, so it's more fun, right? So you can actually kind of move things and decide what testing is needed based on what evaluations are needed, not necessarily testing, based on how your device is used, okay? So these are up here. Maybe more importantly, as I throw that on the ground, if you guys want a copy of this presentation, I'm more than happy to hand them out or to send it to you. I need your email. So if you can just give me a business card with your email or a slip of paper with your email, legibly written, I'll send you guys a copy of the slideshow afterwards. That's no big deal, okay? So today, though, let me introduce myself. For those of you that don't know me, my name is Thor Rollins, and yes, that is my real God-given name. My dad was big into mythology and said my first son, I'm going to name Thor. He didn't know about all those movies that are going to come out to make my life hell. But besides that, it's been great. I love the name. And I've been with Nelson for 21 years. All of that time has been involved in biocompatibility. For the last 12 years, I've been an ISO expert on the TC194 working group, specifically those working groups that write the 10993 standards. And one of those standards I'm an expert on is 10993-1. And so that's what we're going to be talking about today. If you're interested in everything we're changing to the 10993 series, tomorrow we're doing a 3-hour training here at the conference, and it's free, where I'll cover all of the changes that are happening to 10993. We'll go through -1, but we'll also talk about what we're doing with cytotox, what's happening with the new draft of toxicology, -17. We'll talk about -23, in vitro irritation, where we're at with that. So that's tomorrow if you guys are here and want to do more biocompatibility stuff with me. So I'm looking forward to it. But today, this 45 minutes is going to be dedicated to what we're doing in -1. To give you a little bit of update in -1, in 2018, we came out with a new version of 10993-1. And I'll talk, just to kind of ground everybody, if you don't have that already in your vocabulary, we'll kind of talk about high level what we changed there. But I can tell you that we're having a little bit of infighting right now within the ISO group. We have those individuals outside of the US that want to go even more. They want to go more to a risk-based approach. They want to get away from mandating more testing and more kind of evaluating materials and using chemistry to help. Then we have the US FDA that's not too keen on going that fast, that quickly, that direction. So they're kind of against each other. And there was a little bit of a coup that happened about last year or a year and a half ago that came out with this draft. So I'll kind of talk about what happened, where we're at today, what's happening in the future, okay? So that's the concept of the presentation today. So the goal today is to discuss these changes to -1, help you understand what the proposed acceptance of these changes are, where we're currently at in the regulatory arena with -1, and understand what's going to come next. Like, are we going to be forced to do a new 10993-1 for biocompatibility of medical devices? So that's what we're going to be talking about. For those of you that maybe are brand new to Biocomp, welcome. I hope you become converted to how wonderful it is and start becoming Biocomp experts of your own. But biocompatibility basically is a series of evaluations that we use to make sure that the devices that you use on patients are safe. That, yes, that the device helps their life out like it's supposed to, but also doesn't accidentally injure that patient in some way, either through toxicity or maybe some irritation, things like that. There's a lot of things that can go wrong with your medical device testing and with biocompatibility, but most importantly, and what you'll hear me talk about a lot today, is I don't want you to think about biocompatibility as testing. I want you to think about biocompatibility as risk evaluation. Based on your risk evaluation, it determines how much risk you still have, and you mitigate that risk with possible testing, okay? And that might seem weird coming from a testing house, but let me tell you, we love the idea of you guys making more and more devices, and instead of me making you spend all your money and time on testing, okay? So we try to help you get rid of testing by making good risk-based evaluations. And that's what the new Dash 1 is even supposed to do more about. We are going to talk very high level about how extractables and leachables, or chemistry, kind of now is playing a role in biocompatibility, and they want in the new Dash 1 to move more that way. So we'll talk about that too. And then we're going to talk about what the changes and risk assessments going to look like in the new draft. Okay, so for those of you that maybe not understand where biocomp comes from, and this plays an important role in a minute, these are the two kind of Bibles for biocompatibility. We have the ISO 10993 suite, okay, so that's Dash 1, Dash 2, all the way to Dash 23. And then we have the FDA guidance document. And you can think about the FDA guidance document as kind of like the interpretation of the FDA on how to apply 10993, okay? So the FDA doesn't have to follow 10993 like a law. The notified bodies in Europe do. Whatever the ratified 10993 is in Europe, either by the European Commission, they have to follow that ISO standard like it's a law. But FDA gets to kind of interpret how they want to follow it. And so that guidance document is their way to kind of tell you how they will interpret it. The problem is that guidance document actually came out in 2016. It came out in 2016 when I was here at MDM East in 2016. So lots have changed. Lots of things have changed since then. Now, they have reissued this back in like 2021 or something like that, but it's exactly the same. They didn't update anything. So basically, it's the exact same document that was first released back in 2016, okay? So the problem we're seeing is the ISO is actually moving faster, which is ridiculous to think about because the ISO doesn't move fast at all, but the ISO is moving faster than what the FDA has been able to keep up with. So we're starting to see more of a kind of a gap between what ISO wants to do and what the FDA is wanting to do. And hopefully we can close that gap because right now it's getting a little dangerous on how far apart they're getting. Okay, so way back when, the reason why we came out with the new Dash 1 is because lots of people think of biocompatibility as this chart. Okay, this is actually the chart out of the old FDA guidance document. It was called G95. For those of you that have been around long enough to remember this guidance document, it came out in 1995 and was replaced in 2016, okay? But this was how the FDA, actually this is before 2016, this is how everybody did biocompatibility, which is they just followed this chart, okay? The concept is you look at your device, you look at the materials and how long it contacts the body, and then you look at what type of contact that it has on the body, and depending on how long and what type of contact, you just did the testing. So it was easy. I even taught at the time my 6-year-old daughter how to follow this because I thought she'd be a scientist one day. Turns out she's more into books, but that's okay. But anybody could follow this chart. And it was easy. You just follow the chart, and then you call up your CRO and you say, I need to do these tests. And that was biocompatibility. It was a checkbox approach. Well, probably back in like 2011, 12 time area, we at the ISO committee said, we got to get away from having people do a checkbox. We need them to start looking at risks. And one reason was because we started seeing people use materials that we really didn't like, but it passed our tests. So as long as it passed the test, people felt good about it. For example, I know of an implant that happened to buy screws from Home Depot. Now, I'm not saying those Home Depot screws are bad. I'm just saying I'm not sure if I want to have Home Depot screws implanted inside me, right? But the Home Depot, and by the way, Home Depot's a great institution. Please use them. I'm not bashing them. Just don't get your implants from them, right? So what I'm saying, though, is my tests probably aren't predictive enough. In fact, a little secret here, these old tests that we've been running for decades aren't very good at all. So like this guinea pig maximization test is only predictive about 50% of the time. So that's why we're trying to progress the science forward, right? But the concept was that these screws could pass my testing, and then they felt good about putting it into a human. So we're like, okay, we got to get away from the checkbox. Plus, we started getting materials that were different. We started getting things like nanomaterials. We started getting biologics and other types of materials that don't really fit the checkbox mentality. So we decided we needed to get away from the checkbox mentality. We needed to completely get rid of it because that does not understand the materials you're using and the processing you're using. So we decided to go to a risk-based approach. And that's what we came out with this version, the 2018 version. And what we did is we changed the chart. So these slide rules actually have the new chart incorporated in them. And that new chart, which I'll show you in a minute, changed how we look at the chart itself. We also added an Annex B, and that Annex B was guidance on how to do a risk assessment. So we said, you need to do a risk assessment first. Well, we realized that not many people know how to do a biocompatibility risk assessment, so we did guidance on how to do a risk assessment. We also added some new definitions. So this is the new chart, and some of you probably already know this, but just bear with me for a couple seconds. As you can see, there's some things that we changed a lot. Okay, so this is the biggest change to this chart since it was originally written back in like the 80s. So the first thing we did is we added a new column, which is we added physical and chemical information, okay? So this means that you are required as a medical device engineer or OEM to know all the materials and processes that are going into your device. Not just know them, kind of know what kind of risk they may pose to the patient, okay? And so that doesn't mean that, oh, I'm just gonna pick up this polymer because it's, has a medical grade on it. By the way, don't get me started on medical grade. It doesn't mean anything, okay? So don't take that for word. Actually ask why it's medical grade. Anyway, that's gonna take me over time if I get too much down that road. But you need to look at physical and chemical information. Also, we wanted to make sure that this didn't require you to do chemistry on every single device. We wanted to make sure that was the case, so we put physical and/or chemical information. We had some notified bodies right away start to say that this required chemistry on everything. This does not. So please don't think this requires chemistry. But it requires you to understand the physical and information on your materials on your device. Then the concept is, oh, by the way, we still make mistakes at the ISO level. As you can see, that box is blank. It's supposed to say sensitization. We'll get that fixed. Sorry, even us, we're not perfect. We also separated these. I won't get too much, but these are the changes we made in the chart. But the big one was this column that we added, this X. And then everything else you see is E, okay? So what that meant, and you can see it here, X means it's a prerequisite for testing. E stands for endpoints to be evaluated. And that's why I want you to get away from thinking about testing, okay? Don't think about these are tests. These are endpoints that you need to evaluate in your risk assessment. Now, you might need to test. Depends on how much information you have and how confident you are about your risk of your device. But I don't want you just to say, these are the tests we have to perform. So looking back at the X's and E's, as you can see, the only X is that physical and chemical information. So you're supposed to do a risk evaluation, that's required, and then evaluate all the other biological endpoints on this graph to see if they're needed. This was the concept in 1093-1, 2018. Some of the other things we added, so we added Annex B. Like I said, this is exactly what it says in there as far as a framework for a biological evaluation. We're supposed to try to minimize the actual animal test that we run. So one of the things we wanted to do in 2018 was minimize the number of animal tests we run. Just to give you guys a sense, and I say this a lot, not to like make everyone mad at me, but in my 21 years, I have tested over 500,000 guinea pigs for sensitization. How many of those guinea pigs had a failure? You guys, anybody want to guess? 1. 500,000 guinea pigs, 21 years, 1 test failure, okay? So that goes to show you how sometimes asinine these tests are. So how can we get away from this? Are there other ways we can evaluate these endpoints other than just keep on testing in guinea pigs or rabbits? The one thing I want to add here, for those of you that may not be aware, and not many people use this, but this is in the standard right now and we use it a lot, which is transitory contact. We were tired of evaluating devices that have very short contact with the body. So what we wanted was to put in a definition that said if your device has very short contact with the body, that it has no biocompatibility risk. We couldn't agree, we all agreed on the concept, but we couldn't agree on a length of time. So, like, for example, some people like Europe said, oh, if it's below an hour, that should be transitory contact. The FDA said, no, we think below a minute. Well, you're not going to be able to get together to kind of agree if somebody's at an hour and somebody's at a minute. So we ended up just basically saying a very brief duration of contact. But if you do have less than a minute contact with the FDA, you can use transitory contact as a way to justify out of biocompatibility. Okay, so that was the old one. Now the new draft. What's changing today? The reason why we're going through some drastic changes today is because we put that 2018 in and everybody still just went right to checkboxing. So very few people are actually doing risk-based approaches and trying to justify out of testing. And that includes FDA, right? And this is where we're starting to see the gap between FDA and the other parts of the world. So what happened is a group of individuals from Australia and Europe and other places decided that they would kind of go around the system a little bit, and they proposed to update this standard. And when they updated it, they completely rewrote it. So the new draft document that they gave us was completely different from 2018. So that made us like shocked, and you'll see what happened here in a second. But the biggest changes is more of a risk-based approach. In fact, they basically took 14971 and incorporated it completely into 10993. So they kind of tweaked it a little bit to make it like biocompatibility, but really it reads a lot like 14971 now. They also put in a lot stronger language on the 3 R's for animals, which is replace, reduce, and refine. So they're trying to push the standard committees to get rid of animal testing. Remember, -1 is the overall umbrella standard that kind of impacts all the 10993 standards. So if this starts to say you have to look to replace animal tests, that impacts every single individual standard. So there's also more emphasis on what we call a BEP, which stands for a Biological Evaluation Plan, okay? So there's actually a whole new annex where we participated in writing like guidance on how to write a biological evaluation plan. If you're gonna do more of a risk-based approach, that risk has to be in your biological evaluation. And so we actually put in there how to do it. By the way, if you ask any FDA reviewer, the reason why they reject most BEPs is because the person writing the BEP did not put enough information about the device itself in the BEP. Like a lot of times, especially if the company themselves write the BEP, they're so used to that device that they'll just be really quick on what it is. And sometimes you guys use like little code names and things like that for a device, and sometimes you'll use a code name instead of the real name, and it gets really confusing. So if you're gonna write your own biological evaluation plans, please spend a lot of time on describing the device, put pictures in there. Heck, I even recommend sending them an actual device with the submission. Because when I go to my friends in the FDA, I see devices that they've approved on their desks. They take kind of pride in approving those medical devices. So it kind of helps kind of see what it looks like when they have it in their hands. But that's the one part that we see is the biggest impact to BEPs is not a good enough description. So anyway, these are the things that we put in right now in the -1 about BEPs. Okay, so the other thing, this goes to the FDA. So you would think that the FDA would be all on board with this biological evaluation plan. This right here, this paragraph is right out of the guidance document. And I'm gonna use a little bit of a PowerPoint math or dynamics here. You can see how good I am at PowerPoint. Down here at the bottom, you can see that it says material plus manufacturing processes plus intended use of the device equals the plan, okay? So that's right there in the bottom of the paragraph. Basically what the FDA says is that if you look at your materials and you look at your processing and then how the device is used, that's the 3 components in your BEP. So the FDA encourages you to write biological evaluation plans. So it would maybe think that the FDA is behind this whole change, but we have not seen the FDA be very happy with this proposed new draft, and I'll talk about that in a minute. By the way, just so you guys know, out of all my years testing biocompatibility, I've tested the same materials from the same suppliers, thousands and thousands. You guys all use pretty much the same suppliers, okay? What makes a big difference is the processing. What you guys are leaving behind on those materials. When I see failures, if it's the material, I know it's gonna fail because I've seen it fail thousands of times. If I don't know if it's gonna fail, it's because there's something from the process residual left behind, okay? Okay, so physical chemical information, we put a little bit more information about how to do physical and chemical information and material characterization. I do want to kind of hone in just a little bit. I mentioned this before, but there's a big difference about the type of information you get from your suppliers. Previously, you guys would just say, hey, you know, is my material, I'm gonna put it in a medical device, is your material good? And they'll say, oh yeah, we got medical grade, or we got USB class 6, or whatever, so it's all good. And that's where you guys stopped. We've got to be a little bit better than that. We've got to understand what goes into those certifications or those identifications. So some of the things you might want to look for, besides the supplier information on those, is predicate testing. Have they done testing on the materials themselves? And if they did, what kind of testing did they do? Does it meet the endpoints that you're required to meet? Does that help you with your risk assessment? For example, and I don't get paid by NewSeal, so please, I don't even know if NewSeal's here, but NewSeal does a great job of doing master files with the FDA. So they'll have all their medical grade silicone and they'll do biocompatibility tests like implants and systemic toxicities on sterilized material. So they can show you that if you sterilize your device within that same range, that they already know their device has passed those tests. Okay, so you can get that master file information with permission from NewSeal from the FDA, and that helps you with your risk assessment. Other companies might just make that material on its own dedicated line. It's the same material that they give to the airline, to the car industries, but they call it medical grade so they can charge you more, but they just put it on their own dedicated line. There's no difference. So really understanding what goes into those supply information is important. And then if they provided material characterization, that's also a good thing. This is my favorite thing we're adding to the draft. And by the way, just like other US representatives, I did vote negative on the draft. We'll get to that in a second. But this is the thing I was most excited about, which is biological equivalence. So right now in the industry, we're seeing, as never before, more changes in materials. Like your guys' suppliers, I know that we just went through COVID and the freeze down in Texas and all that stuff, but you guys can't get material suppliers or you have uncertainty in your supply chain. So you guys are changing suppliers or getting multiple suppliers for the same materials. And I'm doing all those biological evaluations. And it's a pain in my rear. I mean, it's so much work. But what we're adding is something called biological equivalence, which is, for example, a lot of people think, oh, I'll do chemistry on my supplier, and then I'll do chemistry on my new supplier. As long as the chemistry matches, then I can say it's the same. I can tell you that from lot to lot, the same supplier is gonna have different chemistry. Chemistry from polymers change so much that it's impossible to have chemical equivalence. So what we decided to add to the draft is biological equivalence, which is, can we prove that the change has the same biological impact as the original material? And we give actually guidance in the draft on how to claim biological equivalence. By the way, the FDA is also coming out with a new guidance document on biological equivalence, where they also give direction on how to claim biological equivalence for changes. So in the next probably year or so, we should have 2 really good sources of standards and guidance documents on how to do biological equivalence. So that's something I'm really excited about. Oops, went too far for some reason. Okay, so this is one of the reasons why I voted negative for this draft. They put in the draft individual guidance for each one of these endpoints. For example, for hemocompatibility, in -1, they gave guidance on what to consider for hemocompatibility. For those of you who don't know, we have 1093-4 all on hemocompatibility. The people who wrote the new draft, the -1, their concept was that not everybody can go and afford to buy all the individual standards. So they wanted -1 to give some guidance on every biological endpoint, which I get, but I think it's going to be confusing if you have a little bit of guidance in dash 1 and it maybe contradicts or maybe expands in dash 4 without having all the information. Plus, if we update dash 4, how does that impact dash 1? We'll have to go back and try to update dash 1 really quickly. So I just don't like that concept as far as actually applicable kind of to standards goes. So that's the reason I voted negative, but every single one of these endpoints had individual guidance in dash 1 added to it. Okay, so there was 2 negative votes. Everybody in the United States, there's 5 of us that vote from the US on dash 1. We voted no, and Germany voted no, okay? So after the US, especially when the US votes no, people freak out because that means the FDA is not gonna recognize the standard. So we met and we went through all the reasons why the US didn't like the standard and why Germany didn't like the standard. At the end of that, the US FDA basically said, look, can we just go back to the original draft and work on that? But because it was an ISO vote, and because only 2 countries voted negative, the majority voted positive, and that meant this new draft goes forward. So this new draft that has it laid out is gonna go forward. What we have to do now is work it to a point where the FDA can agree on it. Because what I'm worried about is if the FDA doesn't recognize dash 1, that means you guys are in a spot where Europe's gonna require you to follow dash 1, and the FDA is gonna have its own version of dash 1, and we may have to do 2 different types of evaluations for the 2 different areas of the world. That scares me, because that's more work than I want to do. So hopefully we can get to a point where we can combine it back together where the FDA can actually accept 10993-1, the new draft. The last thing I wanna talk about, and for those of you that joined in the back, if you want a copy of this presentation, I'm more than happy to give it out. Just give me your business card with an email and I'll send you a copy of it. So the one thing that I'm gonna end on, that's the biggest change in the new draft. The people writing the new draft got tired of people checkboxing, and their thought is engineers are always gonna follow a chart like a checkbox. It's ingrained in engineers' brains. If there's a checkbox, they're gonna just checkbox it. Or if there's a grid, they're gonna checkbox it. So let's get rid of the grid. Let's get and make it more of a concept flow. So what they did is they looked at the chart and they said that we could break the chart down into a couple of concepts. First is what we call the big 3, which means every medical device has to evaluate cytotoxicity, sensitization, and irritation. That's the big 3. So we'll just say, hey, you have to evaluate the big 3. Then you can see that there's a repeating pattern within the standard. So they said, look, let's follow this pattern. So this pattern makes sense, and we can talk about what that's in that pattern. Also, there's no E's over here, and that's because we can't come up with a biological kind of device or a for sure contact type that requires it. It depends on how the device is used. So we said, okay, so there's no E's over in the blue spot. So let's put together a verbal flowchart. So this is the actual verbal flowchart that they recommended. And if you look at it, to me, it's a lot more confusing than a chart. In fact, this looks like something where if someone put this in front of me, I'd say, you know what? Let's make this into a chart. It would be much more easy to follow, right? So I don't know how this is gonna end, but basically it says first you do the big 3, and then if you have mucosal contact, you have to evaluate these other endpoints. If you have breached or compromised surface contact, you have to do those. Or if you have direct tissue contact, you add that. And at the end, if you have indirect contact with circulating blood, you add that. So I think that's really hard to understand and follow. So I'm hoping we can get back to the chart and still kind of emphasize that that chart's not supposed to be checkboxed, that you're supposed to evaluate those endpoints. But this is right now how the new draft is written, is this flow chart. Okay? So the main goal going forward, this is by far our main goal. And by the way, the 2 people who wrote this draft said that they knew the FDA wasn't going to like it and wasn't going to accept it. But they said that they could move science forward by going really far one direction and then coming back to the FDA. And that pushes the FDA to go a little bit farther too. So that was their goal, is to try to reel in the FDA this way just a little bit. So we'll see. Definitely nobody wants the FDA not to recognize -1. So we're working as a writing team to rewrite this draft with the FDA's backing on how it might look, okay? So that's kind of the update on Dash 1. Thank you for your time.